to talk about pigment cells development. So we can see that we have a huge diversity of pigment pattern and color hues in the vertebrates. And a lot of what we know today is due to research done in the zebrafish that you see on top left. But huge evolutionary distances separate these vertebrate clades from the zebrafish, and we also have events like loss or metamorphosis that separate these clades. So our idea was to explore the tetrapods' color pattern development diversity. And for this, we use two squamate species, the corn snake and the bearded dragon. We harvest the embryonic dorsal skin, and we use these cells for single-cell RNA sequencing. In the middle, you can see the range of pigmentation from our first to our last time points for both species. And we identified the skin populations that we're expecting, and we focused on this purple cluster here, the chromatophores, also known as the pigment cells. We use this data to reconstruct the developmental trajectory from these cells. So we start from pigment cells progenitors on top. These cells rapidly differentiate into melanophores that give this brown-black pigmentation. And then we have these intermediate stage progenitors that will give rise to xanthophores that have yellow to red pigments, and iridophores that reflects light. We have a bit of a similar picture, but not exactly the same in the bearded dragon, where we actually saw that we have extra populations. So we have these secondary melanophores and secondary xanthophores that share a common transcriptional program. And we also have these progenitor-like cells that express very specifically ASIP, a gene important for ventral coloration in vertebrates. And then finally, what we wanted to see is look at the transcriptional profiles of these cells, of the different pigment cells across species. And what we see here on top is the top 10 corn snake progenitor markers and their orthologs in the bearded dragon and in the zebrafish, and the color is the expression enrichment. And we can see that we don't have so much, we have poor expression of the orthologs in the zebrafish and bearded dragon, but for the melanophores, it's very different. We have all the melanin biosynthesis genes that are enriched in both in comparisons in both species. Very short conclusion. So the pigment cell progenitors show more diverse transcriptional programs, but likely due to functional constraints, the differentiated pigment cells show similar gene expression patterns. And with additional population seen in the bearded dragon, we can see that we have some diversification of these cell types in squamates. Thank you.